Dear participants, on behalf of the Guru Nanak Center for Research, I welcome you all to day 7 of the International Industry Academia Conclave IIAC 2020 on redefining phytomedicines for post-COVID era. Today, 11th of November 2020. Now, I request Dr. K. Namashivayam sir, Head Department of Physics, Guru Nanak College, to kindly welcome the participants. Sir, please.
Thank you, sir. Good afternoon to one and all present here. Respected General Secretary and Correspondent, Sada Manju Singh Nayaji, the Dynamic Principal of Guru Nanak College, Member Indian Science Congress, Dr. M.G. Raghunathan, the Dean Research and convener of this mega international event on redefining phytomedicines for post-COVID era, Dr. J. Jayanti, the eminent resource person of today's session, Dr. A.M. Gunawan Indrayanto, Professor, Surabaya University, Surabaya, Indonesia. It gives me immense pleasure in welcoming you all to the 7th session of the International Industry Academia Conclave IIAC 2020 on redefining phytomedicines for post-COVID era. The International Industry Academia Conclave 2020 is mainly addressing the issues related to the present-day challenges on research and development of medicine industry and its requirements from the present generation graduates, especially from the life science team. Really, it is aimed at narrowing the gap between the availability and requirements. A special mention to be made on Guru Nanak College Center for Research. It is generally thinking globally and acting locally. Now, through the IIAC 2020, it acts globally too. The entry for today's session is more than 1,100 participants across 15 countries. Today, we have with us the eminent scientist, editor of many international journals with more than 120 international publications to this project, Dr. A.M. Gunawan Indrayanto, Professor, Surabaya University, Surabaya, Indonesia. On behalf of Guru Nanak College, I welcome Dr. Gunavan Indrayanto, Professor, Surabaya University, to this international conclave. Welcome, sir. We are very happy and sure that the entire audience will be enriched with new ideas after listening to you all. I welcome all the dignitaries and participants to this mega event. Thank you for the opportunity. Thank you.
Thank you very much, sir, for your warm words of welcome. Now, I request the convener of this IIAC 2020 and Dean Research of Guru Nanak Center for Research, Dr. J. Jayanti ma'am, to kindly introduce our resource person to the participants. Ma'am, please.
A very good afternoon, everyone. I'm very happy to introduce our today's keynote speaker, Professor Dr. Gunavan Indrayanto, who's working as a professor at the Faculty of Pharmacy, Surabaya University, Indonesia. Dr. Indrayanto got his doctorate in 1983 from the University of Tübingen, Germany. He worked as a visiting researcher at Leiden University, Düsseldorf University from 1994 to 2002. He was a former senior professor at the Faculty of Pharmacy at Erlangen University. He is also the former member of the Indonesian National Board of Education, the member of the Research Council of Indonesian Higher Education Board. In 2008, he was nominated as the leading scientist by CONCLAVE at the OIC Standing Committee on Scientific and Technological Cooperation, and in 2009, he was also nominated as an excellent researcher by SALAI EU-NET. He was awarded the Ahmad Bakri Award 12 for Health Sciences 2014 recently, and he was honored the Satya Lenkano Karya Satya 2017. He served as an editor and as an advisory board member of many accredited Indonesian national journals, and to name a few: Journal of Sciences, Indonesian Journal of Pharmacy, Alchemy, Indonesian Journal of Veterinary Sciences. At present, he is also the member of the advisory board of the Journal of Planar Chromatography, Springer, Germany. He is the editor or the contributing editor of some of the volumes of the serial book of the profile of drug substances, excipients, and related methodology of Elsevier. He is also the editor of Advances in Pharmacological and Pharmaceutical Sciences. He has served as invited reviewers of some international journals such as the Journal of Pharmacology and Biomedical Sciences, and also some national journals also. Dr. Indrayanto currently has 120 international publications consisting of original articles, short communication, and review articles. His publications are, as per H-Index, H10 Index, were listed in the Scopus, ResearchGate, Google Scholar, Cinta, and other databases. Presently, his main works are analytical method development, both in chemical and bioassay, and its validation, metabolite profiling, and natural product chemistry. Thank you so much, sir, for having accepted our invitation and spending your valuable time and knowledge with our participants. Thank you very much, sir.
Now I request Professor Dr. Gunavan Indrayanto, sir, to kindly take over the session. Sir, please.
Okay, first of all, thank you very much for this invitation. I am very honored to be present here to share my knowledge. And at first, I should apologize if my English is not so good. Now, I will share screen.
Sir, you have to share it again, sir.
I have difficulty to share. I do not know why.
Yes, sir, but it got closed. You can share it again, sir.
Okay. Close.
No, you can share screen. At the bottom, you'll have the share screen option. You can click that.
I've clicked.
Then entire screen.
Yes.
Then share.
Yes. Hello.
Yeah.
Nothing happened.
Yeah, we got. Now you can open your PPT, sir.
My PPT is, oh, my PPT is, yes, okay, sorry.
Yes, sir. You can make it full screen, sir.
Can you show this slide?
Yes, sir.
Okay, thank you. Sorry, I have mistake click. Today, my presentation is validation method of chemical and biodiversity characterization of phytochemical and herbal drug. This is the title of the presentation. Herbal drugs, sometimes called botanical or phytomedicine or herbal medicine. Herbal derived drug usually comprised of very complex chemical mixture of natural product and often containing multiple herbal constituents. It is very common a single herbal product could contain many herbs or an extract. Herbal drug seems very important because 80% of developing world population use or consume this herbal drug. In Indonesia, around 75% Indonesian consume this various of herbal drug. In Indonesian, herbal drugs commonly called jamu. Jamu has been used for a long time to maintain good health and treat diseases, and standardized jamu preparation consisted of standardized extract and phytopharmaceutical. We know that there are countless scientific publications on herbal drugs, but unfortunately, most of the herbal drugs are not yet fully scientifically established regarding their safety, efficacy, and consistency. This is the reason why the application of the herbal drugs by medical doctor in Indonesia is very limited. Therefore, the research of herbal drug in order to develop into modern drug, I think is very important. Today, I will discuss about this how to develop from traditional drug into modern. This is some example of publication showing the workflow of studying medicinal plant. The number I mentioned here will be mentioned in the next slide. First, always we do literature review and then evaluation the literature and designing the methodology and then collecting the plant. After that, doing toxicity or safety and then bioassay. And after that, we use phytochemical study to do fractionation, isolation, etc. And we develop stable, standardized or compound, and then doing in vitro. And if all okay, we are doing clinical study. This is the method that's published by Professor Suntar 2 years ago. All steps used in chemical and biological assay. This all step here, they use chemical and biological assay. This is some important aspect. Developing modern drug from medicinal plant. The number is same with the number I mentioned before. At first, collecting and authentication of the plant material, and then predicting or determining the possible active compound and their chemical structure using chemical and biological method, and determining the phytochemical that can be used as marker for quality control purposes, determining of the detection limit of the contaminant or toxic compound, and then isolation, chemical structure, determination of the active or toxic compound or unknown, development of standardized and stable herbal drugs, and clinical study. In this presentation, I just present number 2, 3, and 4. To have reliable data of the characterization of the phytochemical, all used chemical and biological method must be first validated. This is what we call it phytopharmaceutical. Phytopharmaceutical is identical with modern drug, chemical drug, but the difference here, the phytopharmaceutical, the active pharmaceutical ingredient is herbal raw material and or extract. That is not pure chemical compound. This is for comparison. Digitalis tablet, digitalis leaf tablet is a phytopharmaceutical, but digoxin tablet is not phytopharmaceutical. Digoxin tablet is modern drug. This is the definition of phytopharmaceutical according to Indonesian Drug and Food Control Agency, number 32, 2019. Phytopharmaceutical is a product that contains ingredient in the form of plants or animal, mineral ingredient, extract preparation, or a mixture of this ingredient, which have been proven their safety and efficacy by preclinical and clinical study. And all raw plant material, extract, and their finished product have been also chemically standardized. If clinical studies are not yet performed, the traditional drug just called standardized herbal drugs, not phytopharmaceutical. We know that the bioactivity or the toxicity of herbal drugs and phytopharmaceutical is dependent on the whole phytochemical content, qualitative or quantitative. I will also to determining how to determine the active or toxic component and its chemical structure. There is two methods. The first method is what we call bioassay guided isolation of active compound. In this method, the raw material will be extracted with certain solvent, for the example ethanol, or we can do it other solvent, and then we should do bioassay to determine whether the extract is active or not active for certain bioactivity. If the extract is active, and then can be continued to fractionation using solvent or chromatography or others, and then we will get some fraction maybe A, B, C, and so on. And each of the fraction should be tested its bioactivity. If the fraction have bioactivity, the fraction can be continued for further work to isolate the compound. If it's not, and then it's not. If from the active fraction using chromatography or thin layer chromatography on after we can purify, then isolate the compound. The isolated compound can be further tested regarding their bioactivity. This is the first method how to isolate the bioactive compound by using bioassay guided isolation. The second one is using combination liquid chromatography, mass spectrum, and multivariate analysis. By this method, we can directly predict the active or toxic compound without any isolation. Something like this, we have LC-MS profile, we have many samples. All the sample measure the MS, and we also measure the bioassay, and using combined multivariate analysis, we can decide whether the sample is active or whether the sample is not active. And by this, from the active compound, we can use the S plot to determine which compound is active. For the sample, this one maybe. That is all. All this is showing the peak of this chromatogram, and then from this we can the number of year we can still test the active compound, and by this we can try to predict this structure without any isolation. But before we can work further, we should validate the method. The method, according to Professor Wan, all quality control sample or validated sample must show a tight clustering in the observed score plot, and the coefficient variance of the described variable should be less than 30%. Variable which show more than 30% should be removed. This is the example. This is a score plot that is show that the quality control sample or certain sample show tight clustering in score plot. It means that the method is valid. If the quality control is not a tight clustering, it means that the method is maybe not valid, and we should try to do again. After we know this, which compound is active, we can proceed to predict the structure using database. This is the steps taken from the LC-MS. We should, from the mass spectrum, we should determine the exact molecular weight based on its adduct. The adduct should be confirmed by extract ion chromatogram. After we have the molecular weight, and then we can determine the molecular formula by using isotope pattern. After this, after we have the MS2, we can confirm the fragment ion based on the observed, and there based on the observed mass spectrum and their extract ion chromatogram, and inserting the MS2, MS, and the molecular formula to certain database. Database can predict which compound probably suitable from this data. After that, we can evaluate the fragment, and we can also evaluate using other database. For confirming the predicted chemical structure of the active compound, we can use authentic standard. This is ECS for unknown, or comparing by published data and or mass spectrum online database. That's all unknown. If unknown, I think we should isolate the compound, purify, and determine the structure using NMR and mass spectrum. This is can illustrate something like this. This is the mass spec, the LC-MS. From the LC-MS, That is, we can select from the peak, we can determine the molecular weight, and we can also determine the isotope pattern. From this, we can determine the molecular formula. Inserting the molecular formula and the MS2 into certain database, the database will predict, we can predict one or two or more compound, and after that we should confirm whether the prediction is okay or not. For that purpose, if we have authentic standard, that's very easy. We just do MS again, standard and our compound. If that's identical, if the mass spectrum is identical, and that is, we can confirm the structure. If it is not, we should do with compare with publication or other mass MS database and evaluate it using an official guideline. For confirmation the peak identity of known compound, usually the best method is using the guidance of Commission Decision 2002-657 from European Commission. With this guidance, the identity of a peak can be confirmed if the peak has identification point at least 4, and the ratio of the daughter ion fulfill this table. For LCMS, the identification point is 1, so it means for LCMS it needs 4 specific fragments to confirm the identity of a peak. If we have LCMS-MS, if we have LCMS-MS, we just need one precursor. The IP is 1, and the daughter, the transition product is 1.5. The total IP is 4. So if we have LCMS-MS, if we have one precursor and 2 daughter ion, we can confirm the identity of the peak. But if we have high resolution mass spectrum, we just need one precursor. The IP is 2, and the daughter ion is 2.5. The total is 4.5. The best accuracy should be less than 5 to 10 ppm. We know that every organic compound have specific molecular ion and fragment. We can trace each of compound in Google to see what is the molecular ion and their specific fragment. As every compound we can search in Google or other database. This guidance from the FDA 2015 just show the same method. For the example here, if we have only mass spectrum, the minimum fragment should be 2. So it's been the IP 2 times 2 is 4. Because this is high resolution. High resolution, the value is 1 ion is 2. If MS, we need also 2 fragments, the IP 5, and we have combination mass and mass MS1 and MS2. We just need one precursor and one daughter ion as described before. The IP is 4.5. The difference is with this guidance. The extract ion chromatogram of the standard and the extract ion chromatogram of the sample should be identical. This is not more than 2.5% or 1 minute. This guidance is not available in this Commission Decision number 657/2002. This is the best, I think. If we use this method, we can confirm the identity of any peak of known peak without any isolation, and without standard we can confirm the identity of the pig. Now I continue with determining of phytochemical that can be used as marker for quality control purposes. I will just only describe the most important aspect of the validation method. Ideally, according to Professor Ponsusan, ideally chemical marker for the purpose of quality assurance or quality control should represent the chemical identity profile and or reflect relevant pharmacological and or toxicological profile of the herbal medicine. Unfortunately, there are currently no detailed accepted official guidelines which can be used to select chemical marker. Challenges in assuring quality of herbal product are the phytochemical primary and secondary metabolite composition in herbs is very complex. That's why chromatography is the method of choice of analysis. The second problem is many external factors can affect the biochemical content, qualitative or quantitative. We know that some external factors such as plant species, variety, site of cultivation, age of plants, kind of organs, season and time harvesting, handling process after harvesting, method of drying, method of cutting, number of herbs, extraction method, formulation, fabrication, and so on. This can influence the content of the biochemical content in the herbal. This depends on the site of cultivation. So it means that identical herbs could have different bioactivity due to different content of their phytochemical or metabolite, qualitative or quantitatively. Due to variation in raw material, harvesting, extraction, and formulation as described before previously, assaying of a certain phytochemical or marker could be not always applied for indicating the quality or safety of the herbal drug. To overcome this problem, Ben Susan in 2015 proposed a ranking system of marker that could be used for quality control purposes. That's called Herbal Marker Ranking System. Every marker will be ranked as X, 0, 2, 1, 3, 4, and 5. This is the ranking system of Professor Ben Susan. If the marker X is mean there are no bioactivity studies currently available for this compound. 0, compound activity is not related to indication of the disease of interest and or no pure reference standard is available. Rank 1 means compound bioactivity is indirectly related to symptom or disease or compound is present in too low concentration in the herbs for it to be screened below 5 microgram per gram, or the compound activity is not supported by traditional or therapeutic use of the herbs. Number 2, compound has bioactivity related to a symptom, major or minor, of the disease. However, there is only one good quality study to support this action, and compound is present in low concentration between 5 to 50 microgram per gram in this herb or the finished product, and the traditional or current use of the herb is consistent with the bioactivity of the pharmacological effect of the compound. Number 3, compound has bioactivity related to symptom and so on. That's the difference with number 2. This is the only the concentration higher, 50, and there is only one good quality study. This is the same with number 2. This is the difference only the compound is present in relatively high concentration. Number 2, this is from 5 to 50 microgram per gram. Number 3 is above 50 microgram. This is the difference. Number 4 is all equal with number 3. The difference is only there is 2 reported good quality study. all the same. Number 5, same to 4, but the compound has the metabolite, the compound and 8 metabolite are bioavailable. It means that they have already doing pharmacokinetic study. Or number 5, compound may be very toxic, needs to be screened to comply to safety limit. It's usually used for genotoxic or carcinogenic, where the maximum use per day is 1.5 microgram. The mean of quality study is here is both adverse and clinical study. 3, 4, and 5, that is 2 report of clinical study. In 2017, Professor Langer published in Plata Medica. I have a summary of this publication. They wrote, nearly all monograph on herbal drugs or herbal drug preparation in European pharmacopoeia contain an obligatory assay of a certain compound or a group of compounds and define a minimum content. We know that if we open Indonesian pharmacopoeia or USP for the herbal, there's just only minimum. Generally, maximum content was not described. This also described in USP 2020, Indonesian Pharmacopoeia 2070. This is different with chemical compound. If chemical compound is always, there are a maximum and minimum content of the drug. In most cases, that obligatory, the minimum content, is inappropriate for the herbal drug evaluation, as the assay constituent must be considered as purely analytic marker without correlation to their efficacy and safety. So it is nice to initiate scientific discussion in order to adapt current quality standard for herbal drug and herbal drug preparation to reflect current scientific knowledge and requirement that are appropriate for quality control of herbal medicine. So the marker should be correlated with its pharmacological action. This indirectly, the publication of Prof. Langer is confirmed what have Professor Bensuson wrote in the publication in 2015. In the same time, research from China, Professor Liu and Professor Wenxi described what it called quality marker. Quality marker, according to them, can be defined according to the following condition. Quality marker should be existing in herbs, extract, herbal product, and formulation. Quality marker could be analyzed using qualitative and quantitative approach. It can ensure the geoauthenticity and for differentiating the quality of the some herbs. The drug effect, such effectiveness and safety, should be demonstrated to be associated with their quality marker. The quality marker is a chemical substance that is traceable in the whole process of production and preparation. If we compare this definition and we compare with the definition of mass of marker ranking system of Professor Bensuson, this is about the same. So it can be concluded only quality marker or herbal marker ranking system 3 to 5 can be applied as a quality tool for evaluating the quality of herbal drugs. The method of analysis of key marker should be first validated. If key marker is not yet specified, it is recommended to apply metabolite profiling combined with multivariate analysis as a key tool. For doing the analysis, we should have validation method, and this is validation method. The parameter is selectivity, linearity, accuracy, precision, range, detection limit, quantification limit, robustness. I have 2 publications that summarization on the recent development of this validation. You can send me email. I will give you the full PDF of these two publications. In this presentation, I will not discuss all parameters. I just will discuss only selectivity and detection limit and quantitation limit. As I told before, due to the complex nature of herb and plant, chromatography is the method of choice. The important thing is to prove the selectivity. To prove the selectivity of the method, it means that the identity and the purity of the peak should be confirmed using spectroscopy or mass spectrum. We know that identical retention type or identical RF does not mean identical compound. One peak does not mean a single compound. For this purpose, to prove the identity or purity of the peak, we should make UV spectrum or mass spectrum on the target peak. Something like this. If we use PDA, this is the peak, this is impure peak, and this is the pure peak. They have identical UV spectrum. With using a modern SPLC or modern TLC, it's just only simple by click the method. I think all the equipment have software to have software for analyzing whether a peak is pure or not. The problem is different compounds maybe have identical UV spectrum. So I think mass spectrum is a better detector, but mass spectrum is the best. No, because in many guardians and many pharmacopoeias, there's always described minimum of total phenol, minimum of total flavon, and so on. I will show you the latest publication that doing validation of spectrophotometer method for the determination of total polyphenol and total flavonoid. Spectrophotometer method for the determination of total phenol and total flavonoid were generally used because they are economical and simple, but not specific. The important thing is the obtained result were different for both of the method, which use different polyphenol flavonoid standard. And all the difference could be statistically different. So in this case, for determining the total phenol or total flavon, usually we use certain polyphenol standard or certain flavon. The important thing is to select what standard can be applied. Total polyphenol and flavonoid in sample must be determined according to the standard that is found in most abudas in that sample. That is why selecting the correct standard is crucial. And last but not least, the method should be validated with the following parameter. We should determine the linearity, accuracy, precision, range, and so on before we can use the method for routine. Although the method have already published in journal, we should do the validation method. Because the condition of the laboratory of our laboratory and the laboratory of the author may be not the same. Okay, now I continue to determining of sample detection limit. This is very important because if we are working with herbal drugs or phytopharmaceutical or related product, we should show that the product free of alpha toxin, pesticide, elemental impurity, residual solvent, toxic metabolite, and so on. So we should determine the detection limit or quantification limit to ensure that all the herbal samples do not contain this compound. Method of determination and the acceptance criteria of 1 to 4 have been described by USB Herbal Medicine Compendium. You can free download and see this method. Negative means that the concentration is below the detection limit. The detection limit must be below maximum permitted limit. The detection limit or DL is depend on the equipment that we use. If we use mass spectrum, it's be more sensitive. If we use UV spectrum and so on. For determining the LOD or detection, the best method is using the USP 43 2020 chapter 1210. We should make something like calibration curve with low concentration. It is, we can use estimate this concentration by using LOD, LOD by sample method, by using signal for noise. We can do calibration, and this is the limit of detection. This is the formula of limit of detection. We can make this formula in Excel, and we can calculate. For quantification limit, we replace this T with 10. This is the example, and the LOD, something like that. We should determine for each compound, toxic compound, for each of equipment, how low the detection limit. After we know the detection limit and quantification limit, we should determine the sample detection limit according to VELACHERY number 49, FDA 25, 2015. In this method, after we get the limit of quantification from the standard as I described before, and then we can spike the sample that we want to analyze with concentration LOQ, and then we do 7 times, and we calculate the standard deviation, and the limit of detection or detection limit of the sample can be calculated using this formula. It means that each of sample, each of metric maybe have different limit of detection. If we calculate, if we measure the detection limit in leaves, will be different if we measure detection limit in root, and so on. If we measure the detection limit in blood, may be also different if we measure the detection limit using urine. The last one is I will talking about validation of biological assay. The bioactivity of extract or isolated compound can be determined using biological assay or bioassay. This is summary. I have already written this summary of validation in vitro bioassay, but this chapter is in press, not yet published. In this presentation, I just select some important aspect, not all. This is the definition of what is bioassay. This is many definition, but the important is the aim of performing a biological assay or bioassay is to determine the effect of a substance on a certain type of living matter or biological test system. Or biological test system. The definition could be different. If the author is different, you can see this is the definition of USP is different from Lilly, also maybe different from European pharmacopoeia. But the important is for the aim of performing biological assay or bioassay is to determine the effect of the substance on a certain living system. The step should be taken if we want to performing valid in vitro bioassay. I have many review articles, I have many review original article, nice paper regarding activity of certain compound, regarding certificate activity of certain plant, but unfortunately they do not report the validation method. So if it's not report the validation method, we cannot confirm the validity of the data. For performing validated method in vitro bioassay, of course, first we should do literature study for selecting the bioassay method, and then we can compare if we have in our laboratory the cell system, the equipment. We should select the equipment or cell system that we have in our laboratory. And then after that, we should evaluating the stability of extract or isolated compound in the selected solvent, and then testing the biosystem performance using positive and negative standard. After that, if it's okay, testing the performance of the test material, and then determination of EC50, determination of selective index, and determination of relative potency of sample. This is very important if we want to do in vivo or clinical trial. The important thing is all used cell line should be first authenticated. All used cell line, although we buy the cell line, you should first authenticate it. Some test methods have been recommended by NTTC. For example, morphology check by microscope, growth curve analysis, species verification, and so on. After the cell line is okay, it means that we can use this cell line. We should test the stability of our sample or extract in the selected solvent. The sample or test sample, the extract or extracted compound should be stable. For evaluating the stability, usually chromatography is the method of choice. And then we can do dose response curve. Usually they will form 6 point something like this. This is concentration, this be maximum effect, this is minimum. If we're doing biological assay, the curve should be something like this. There is a minimum and there is a maximum. For evaluation the system, the cell, we should calculate the Z factor, standard and sample. Evaluating of biosystem sample performances can be performed by using authentic standard and sample. Before we to determine the Z factor, all data should be checked whether there is outlier or not. Should be checked whether the data described here is outlier or not. Outlier data should be removed. This is, we can do something like this. This is the maximum. This is the minimum. This is the minimum variability. The Z factor can be calculated using this equation. This is variation factor. This is the maximum data. This is minimum, bottom, minimum, negative. This is negative response. We can use negative standard, or we can use the positive response, positive standard. This is from this, we can do something like this. We can calculate the Z factor with this following equation. Also the signal window, also the variability using this formula. And this is the acceptance criteria. What is mean? If we test our test system or biosystem using certain standard, but the value of Z factor is very low, it means the system is not suitable for our purposes. We should search another system, or the system is not okay. We should try to find the system until the Z factor is something like this, acceptable, is between excellent, more than 0.5, and or if the data is below negative, is unacceptable. If Z factor is not acceptable, other biosystem must be tried or to discontinue the works. After using standard is okay, we should do the same thing with sample. The sample also should have a Z factor in acceptance criteria. If it's not, it means it's no use to further to continue the work. And then the important thing is selective index. Selective index can be defined as the ratio of the toxic concentration of the sample against its effective bioactive concentration. The ideal drug should have a relative high toxic concentration, but with a very low active concentration. Evaluation of SE value SI value for any research on herbal drug or isolated compound is very crucial for determining whether the further work can be continued. This is the formula, the equation. This is the equation. SI selective index should be as high as possible. 10 is nice. Below 1 is not acceptable. If we have 1 to 10, we should do experiment for confirmation. Although the extract or the compound is very active with low concentration of IC and MIC, but if the selective index is low, it's no use. It means the sample is too toxic for animal, too toxic for further work. So other biosystems should be tried, or the work need not to be continued. This is the selective index. For continuing the work for in vivo and clinical studies, the accurate potency of the tested sample must be determined. This is needed to decide the correct administration dose. For this purpose, I recommend you to apply the method that described by European Pharmacopoeia 10. This is very detailed. It's complete with equation. We can just use Excel to do the calculation. But we can do also using other official method from the USP 43, General Chapter 111 or 1030 or 1033 or 1034. But I recommend you this European Pharmacopoeia because they write the complete equation. We just only insert the equation in Excel and then we can do calculation. And this is the important thing. If the drug is modern, it means the modern drug should have consistent pharmacological activity. It means batch to batch, they have consistent activity. Because as I told you before, that the problem of using herbal drugs, the content of phytochemical or biochemical can be affected by many external factors. To have consistent pharmacological activity can be evaluated by using key marker. This can be only achieved by evaluation of each key marker. Concentration of key marker in the preparation must be included in the specification ring as described in the analytical target profile. If we have consistent pharmacology action, if we have consistent pharmacological effect, so I think the medical doctor will use this herbal drug. If drug to drug, I mean batch to batch, have not consistent pharmacological action, that's impossible. We ask the medical doctor to use the herbal drugs. For the conclusion, the chemical and biological characterization of phytochemical are very important in herbal drug research. For having reliable data of all experiments in herbal drug research, all chemical and biological methods which will be applied must be first validated according to the newest guidance. If the use method were not first validated, the conclusion could be wrong or biased. I would like to thank to Dr. Jayanti, the Dean of Research, Mr. Raghunathan, Principal, Mr. Sadar, General Secretary, and also to Dr. Mahendra for this invitation so I can share my knowledge in this presentation. I would like to apologize if my English is very bad. I hope you can understand. Thank you very much for your nice attention.
Thank you very much, sir. So we have a few questions in the chat box.
Okay, I will stop this. Should I stop?
Yes, sir, you can stop presenting. Sir, Satya RVS asked, what types of samples can we use for bioassay?
Sorry?
What type of samples can we use for bioassay? I have popped up, sir, in the screen.
We can, for bioassay, usually for determining the activity, usually at first we should extract. If the extract is active, we can continue for isolation. So the bioassay can be performed using extract and or compound.
Dear participants, you can post your questions in the chat box so that it can be answered by the speaker. So I think your presentation was very clear. Everyone has thanked you and appreciated your presentation, sir.
Sorry, maybe my English is too bad, so most of the participants do not understand what I'm talking about.
Certainly not, sir. Too clear. Too clear.
Ma'am, shall I propose the vote of thanks, ma'am?
Yes, sir, you can.
Thank you. Thank you very much, ma'am. Dear participants, I feel very happy to propose the vote of thanks on the day 7 of the International Industry Academia Conclave IIAC 2020 on redefining phytomedicines for post-COVID era. On behalf of the Guru Nanak Center for Research, Guru Nanak College, Chennai, I wish to extend my gratitude to our today's keynote speaker, Professor Dr. Gunavan Indrayanto, is professor at the Faculty of Pharmacy, Surabaya University, Surabaya, Indonesia, for readily accepting our invitation and sparing his valuable time with us in the midst of his hectic schedule. Sir, we are all impressed by your knowledge in the plant medicine, isolation, characterization, and drug validation. I thank our college management, especially our most revered General Secretary and Correspondent, Sardar Manjit Singh Nayyar, our respected Principal, Mr. M.G. Raghunathan Sir, for their constant support, guidance, and help for all our endeavors. I thank Dr. Namachwayam Sir, Head Department of Physics, Guru Nanak College, for delivering the welcome address for today's session. I thank Vice Principal Students Affairs, Vice Principal Shift 2, Deans of all schools, heads and faculty of various departments of Guru Nanak College for their support and for all the center's activities. Last but not the least, the participants from across the globe for your active participation in today's session and in all our programs. Once again, I thank one and all present here. Thank you very much.
Thank you.